In this lecture, we're going to be talking about molecular genetics. We're going to be studying the structure of DNA and how DNA gets replicated. We talked about that in the last unit. And we're going to talk about how your DNA codes for your traits, or how does it code for the proteins that, give you a different eye color, or make your hair curly versus straight, or, causes a genetic disease or doesn't cause a genetic disease. So the first part of our notes here that we're going to talk about on page 1 is just the structure of DNA. And then we'll get into how the structure then leads to these traits that you have. And we've already talked a lot about DNA. DNA can come in different forms. Whenever we see it like this, then we say that we have a chromosome. And remember, when we see it as an X, it just means that the DNA has been copied. But whenever it's all coiled up, it's a chromosome. When it's uncoiled, though, we say DNA is in chromatin form. So let's indicate that. DNA is found in different forms. Chromatin is how we describe DNA when it's unwound. I like to think of this eye as being like a long string. Let's kind of show like this is what chromatin looks like. And this would be chromatin here in that diagram. And then when it's wound up or condensed, we say that we have a chromosome. So chromosome is really just coiled up DNA. Sometimes we'll see DNA and it'll be shaped like this, and they're just unfolding it basically so that you can see what DNA is made out of. So it comes in, different structures, but it's all DNA. Now, DNA sometimes is shown in chromatin form, and it looks like it's wrapped around. Let me focus real quick. It looks like the string is wrapped around these little spools, kind of like thread around spools. And those proteins have a name. They're called histones. When DNA wraps up and coils up into chromosomes, it's easier to wrap it up if you wrap it around something. So that's what these little circles are. Each little circle is a protein, which is called a histone. And then that DNA wraps up, and you can store a lot of it in a smaller space when you have it wrapped up. And that's how it's able to fit into a nucleus, something microscopic that can have almost 6 feet of DNA in it. Now, DNA is a polymer. Poly means many. So that means it's made of many small molecules bonded together, strung together. We've already talked about that. Those small subunits that are repeated over and over again are called monomers. So nucleic acids are made of monomers called nucleotides. That is a review from the biochemistry unit. Now, each nucleotide has 3 parts to it. One part is called the phosphate group. Sometimes people just call it the phosphate, but technically it's called the phosphate group. And then we have another part that is the sugar. And in DNA, the sugar, that D right here, so let's indicate D, and then underlying D here is deoxyribose. That's the name of that sugar. Sugars end in ose, so deoxyribose is the sugar in a nucleotide. And then the third part of the nucleotide is a nitrogen. And watch what I'm going to put here. I'm going to put nitrogenous. It's just another way to say it's got nitrogen in it, a nitrogenous base. Okay, so we see nitrogen atoms in bases. And if you remember, we have carbon, hydrogen, and oxygen are in sugars. So the sugar will have C, H, and O in it. And then the bases have nitrogen in them. And then we have a phosphate group that has phosphorus in it. So that's where this chopping comes from. We learned that those are the elements that are found in nucleic acids: carbon, hydrogen, oxygen, phosphorus, and nitrogen. Now let's kind of indicate here on this picture of DNA. Again, this is DNA shown like as a ladder, so you can see the different parts of it and the bonds. Let's indicate where these 3 parts are. So I have them numbered up here. This P right here is the phosphate group. And then we have this S is standing for the sugar. So that's the sugar in a nucleotide. I'm just putting number 2 there. And then this third part right here is a nitrogen base. So collectively together, let's box around those 3 parts. That would be a nucleotide. So let's indicate that this is one nucleotide, okay? And then there'll be another nucleotide. And then we have another nucleotide, and so on. We also have nucleotides, phosphate, sugar, and a base on this side as well. Okay, so we can see that when we string together nucleotides, we end up with a polymer. In this case, it's DNA. Now, our bases, you can see that our bases pair up. And every time you have a G, you always have a C. Every time you have a T, you already have an A. Those are the 4 nitrogen bases in DNA, and it's these bases and the order of them that gives you your different traits. So our bases occur in pairs. So I'm just going to write A goes with T. You might remember that the stick letters go together and then the curvy letters go together. I'm going to go ahead and write out the names so you know what these words are when I use them in class. So adenine always goes with thymine. And then guanine always goes with cytosine. They all end in this INE. Now, we can see that they're held together by bonds here. So we have some dashes there that show a bond in these different areas. So those bonds are called hydrogen bonds. We've talked about hydrogen bonds before. So we have these hydrogen bonds. We're going to mention them later on in other lessons because we're going to separate the bases by breaking the hydrogen bonds between them. All right, now let's take a look at our DNA here, and there's a few other parts to it. So notice that we have a label here, and it's called the sugar phosphate backbone. Because whenever we look at DNA, the bases are on the inside of it, and then on the outside, which we call the backbones, because it kind of looks like vertebrae backbones that go in and out, in and out. This is called the sugar phosphate backbone because it's just the sugars and the phosphates all bound together of these different nucleotides on both sides. So that's what we mean by the backbones of DNA. And those bonds are pretty strong bonds. So usually DNA doesn't break here at the backbone, but we will see it's much easier to break those bonds between the bases. We can see that DNA is double-stranded. You have a strand of nucleotides here, and then you have a strand of nucleotides here. And notice that they're just upside down from one another. Now, this side isn't identical to this side. If it did, we would say they're identical, but they're not because this side has A on it, but this side, this nucleotide has T. So we say that the 2 strands are complementary to one another. They're complementary. So DNA is made of 2 complementary strands. Just like what goes with peanut butter, and you automatically say jelly, right? If I say what goes with adenine, you would say thymine. So those are the complementary base pairs. They are what always go together. And since this strand is running upside down from this strand, they're parallel because they're running parallel. But since one goes in the opposite direction, we say that they're anti-parallel. So 2 strands of nucleotides that are anti-parallel to one another. So if you know your base pairing rules and I say identify the complementary strand of nucleotides or the complementary bases that would be on the other side of this strand of nucleotides, you just know your rules. A always goes with T. T goes with A, C goes with G. And when we abbreviate DNA, sometimes we just write the bases because the phosphates and the sugars are the same all the way down. But A, G, T, A, C, G, it's the order of these bases is going to lead to different variations and different traits between organisms. Now, notice that the ends of our strand, so here's a strand of nucleotides, here's a strand of nucleotides. The ends look different. So this one has a phosphate sticking out, and this one has more of the sugar sticking out. We have names for these ends. So this end that has a phosphate sticking out is called the 5'. That's like a little apostrophe, called the 5' end. The part that has the sugar is called the 3' end. And then if you look up here, you can see that this phosphate end is sticking out. So that's the 5' end. And then here's the sugar sticking out. So that's the 3' end. Now, those are important. We're going to have to reference them. I'm going to go ahead and put a circle around each of them so that we note what the ends are called. Something to help you remember that. Phosphate and 5 start with the same sound. So anytime you see a phosphate as an ending, that's the 5' end. So phosphate and 5. I have that written right here for you. And then again, that sugar is the 3' end. Now, DNA actually has a twist to it, and that shape has a name. It's called, we see that DNA is a double helix. And it took several scientists working together to determine what DNA was made of, how these parts were arranged together, and then also this final shape that we see here. And so again, that shape we call a double helix because it's double strands of nucleotides, and then they're twisted into this helix shape. And this was determined by a woman who was named Rosalind Franklin. She used X-rays to figure out that DNA has this twist to it. If you look kind of right here, this is the picture that she took and I show in class. So she took, imagine this being a photo, this part right here, and she saw this X part. And that made her realize that there's actually two strands to it and it's twisted. So she was the one that discovered the double helix shape. And that was the final bit of information that these researchers needed to put together what this whole model of DNA looks like. And so the people that do get the credit for determining the structure of DNA is Watson and Crick. But there's a little more to that story, and it was really highly competitive. And they wouldn't have been able to figure that out if it wasn't for Rosalind Franklin. And her information that she found there was taken, and it was unknown for her, to her, and was given to Watson and Crick, and they beat her to publishing. So there's a little more to that story than just, crediting Watson and Crick with the discovery of the structure of DNA. And that's pretty much it. That's just going to be our structure of DNA notes.